All right, thank you so much, Tim. And I'd like to thank also Sherry for this invitation. It's a great meeting. There you go, perfect. So what I'm going to do today is to tell you about some of the studies we've been doing in the lab in the past few years on the circadian clock. And I realize that this audience is not necessarily into circadian biology. So I'll give you some little introduction here. Yeah, what we basically found is that through our studies in the circadian system, is that we were able to establish a direct link between the epigenome and metabolism. And we feel that this part should be of interest to this audience. So circadian biology, the word circadian comes from the word Latin circadian, means about a day. So these are 24-hour cycles that we all have, extremely conserved all the way from cyanobacteria to humans. Plants have it, and insects have it, and so on. It's one of the most conserved systems in biology, evolutionarily, also conceptually conserved. Now, we all feel it. You know, that's what is dominating our physiology, our metabolism. We all have our cycles of sleep and wake cycles. We all have our cycles of feeding behavior or motorial activity. You don't want to go and run in the middle of the night, or you want to have a huge meal too early in the morning or whatever. So basically what happens is that those rhythms really dominate our day-dark cycles all the time. Now, what we like to understand is how they work, and been working on this for several years now. So when we're going to look what is oscillating in our body, basically everything. Hormonal levels, neurotransmitters, even some cell types oscillate in a circadian manner, so a 24-hour cycle. How that happens is because we have an internal clock, and that internal clock is independent from the light-dark cycle, which is outside our classical day-night rhythm. And I'll tell you more about this. Now, when things go wrong in that clock, things can go really wrong. That is, when that 24-hour cycle is disrupted for some reasons, you do have remarkable pathophysiological problems, such as depression, obesity, sleep disorders, neurodegeneration, cardiovascular disorders, and inflammation, even cancer. Suffice to say that, let's say, ladies who work a night shift, like nurses, they have something like 3 to 4-fold more chances to get breast cancer, just to give you an idea. From a molecular point of view, the work of the past 15 years has unraveled the machinery that is dedicated to regulate all our circadian biology. And that's mostly a transcriptional machinery, including genes called clock, BMAL, PER, and CRY, and so on. These are transcription factors organized in the feedback loops, which you'll see in a minute. And these molecules control a large number of what they're called clock control genes, or CCGs, and we'll talk about this too. Now, how this really works is something that we've been trying to unravel from a molecular standpoint. We all have this pacemaker. That's the internal clock. That is a 24 hours based. That pacemaker is independent of input signals and output signals. It will work no matter what. However, input signals are able to reset that clock and to adjust it every day. And the most important input signal is, of course, light. We all feel that way. Let's say I travel to Europe, unfortunately, kind of often, and that makes me a 9 hours jet lag with California. When I get there, my light-dark cycle in Europe is different from what I'm expecting to have. It's expected to have a night instead I got daytime. But that daytime allows me to reset my own pacemaker, and that is readjustment of the jet lag. Now, what light really does is the following thing. Basically, the light signal will hit the retina, and through these ganglion cells in the back of the retina and through the retinal hypothalamic tract in our brain that you see here, will hit a group of neurons, 2 small nuclei, right here located in the hypothalamus, These are called the suprachiasmatic nucleus. There are about 20,000 neurons total, but these neurons have the amazing capacity of oscillating no matter what. You can take them in culture, and they will oscillate with the 24-hour cycles no matter what, basically forever. This is the central hour mammalian clock. And the molecular characteristics of this neuron have been unraveled and still studied in many laboratories. When light hits those neurons, the light will start a whole new program of gene expression that you can see, for instance, here with the expression of a clock gene called par1. And you see the specificity of the system because only those neurons respond to the light signal, no other neurons in the brain. And that system, that new program of gene expression, is basically responsible for the resetting of the clock to a new light-dark cycle or to a new system. That's exactly what happens to you if you go and have a glass of water in the middle of the night and you turn on the light. 5 minutes of light, simply 5 minutes of light, will do this to your suprachiasmatic nucleus neurons. Now, from a molecular point of view, those molecules are organized, as I told you, in the feedback loops or regulatory loops. And I'll show you here a little movie that will help you understand the way things go. This is the sun, giving you the idea as a daytime. These are good guys, B myelin clock transcriptional activators. There's blue and red, and green boxes. And per and cries are the repressors. Now, the activators will transcribe the repressor genes in the proteins. The repressors will be now accumulating into the nucleus. Of course, there are several regulatory levels here. The transcription, the translation, the dimerization of these repressors, and their translocation to the nucleus. Once they accumulate in the nucleus at a certain level, and it's nighttime now, this is the moon, the repressor will shut off their own expression, as is shown here. And by doing so, they block transcription of their own genes. Now, of course, the proteasome is involved in degradation of those repressors, and there the proteasomes and specific F-box proteins are involved. Now, the timing of this process is critical to allow new transcription in a new cycle, and it's daytime again. So all this works in this feedback loop that work over 24-hour cycle. Now, an additional concept I need to introduce to you is the fact that we have clocks everywhere. It's not just in suprachiasmatic nucleus. Work in the past 10 years in the zebrafish, in the fly, and in mammals has shown that we have basically clocks everywhere. In the spleen, in the heart, in the liver, and even in fibroblasts. We can take fibroblasts, the same cells that we've been hearing up to now, and take them in culture. And those fibroblasts have been immortalized for 20 or 30 years. They have a circadian clock, and they will oscillate with circadian biology and responses to the 24-hour cycle. Now, how the suprachiasmatic nucleus in our hypothalamus, in response to light and adjusted by light, will talk to the peripheral clock is a critical area of interest because critically we'll understand much more about endocrinology and neuroendocrinology, understanding what are the humoral signals that will allow the talking from the SCN to peripheral clocks. This is really what we'll say an orchestra director that is required to synchronize all the metabolism and physiology in our body. Likewise, how much motorial activity and feeding activity and feeding functions will feed into the peripheral clocks and back to the central clock. All those are really critical events. And I just want to tell you this to make you understand how important it is to understand the physiology of this system and how much we're going to link now in the rest of my talk, telling you about my data, the epigenome and to metabolism to this system. Now, the very first question that came to us is, let's go and look how many genes are regulated by the circadian clock. And this work done in a number of labs in doing microarrays in the mammal, in mammalian cells, or in fly. Something like 15%, at least 15%. Joan Hagenash at UPenn believes even more, maybe 50%. But 15%, at least for sure, of all our transcripts are oscillating in a circadian manner. Well, that is spectacular if you think about it. Now, it's spectacular for several reasons.